In this video tutorial, I am going to discuss about important exam question, that is telomeres and telomerase are involved in an aging process justify. So to understand this, let's first understand about the telomeres. We know that human genome is a linear DNA, right? And this DNA is organized into the chromosomes. So let's say this is one of such piece of DNA from the human. Let's say this is the 5' end and this is the 3' end. So opposite strand will become 3' to 5'. Now in this DNA, this central part of DNA contain all the necessary genes. So it is called as a coding region. Whereas this outer part and this outer part, you can also call it as a terminal part, it contains a non-coding DNA. So it is a non-coding DNA. That means it doesn't contain any necessary genes. Second important thing is that it contains a repetitive sequence of nucleotides. This nucleotide, particularly TTA, GGG, these 6 nucleotides are repeated thousands of times in this terminal part of DNA's 3' end. So this is the 3' end. So over here there is a TTA, GGG, again repeated same, that is TTA, GGG, and such repetition is around thousands of times. Similarly over here also this 3' end, so TTA, GGG, again repeated TTA, GGG, and similarly thousands of times here also it is repeated. And it is obvious that on the opposite strand the complementary to this nucleotides will be present. So these are all complementary nucleotides. Here also there are complementary nucleotides. So remember this particular sequence, it is found at the 3' terminal of each DNA strand. So this non-coding DNA, repetitive sequence of nucleotides which is found at this terminal part of DNA, they are called as a telomere. Now one natural question arises that this is a non-coding DNA, that means it doesn't have very important piece of DNA. Then why this telomere exists as they are not coding, right? That is a natural question. So the answer is that this telomere has two very important functions. What are these important functions? The first important function is that these telomeres, they serve as a protective cape. Now let's try to understand this. So for that, suppose this is the natural DNA and let's say this DNA is damaged. So what happens? There is a breakage of DNA, double strand break is there. So now this DNA will look like this. There are two pieces. Now once there is such broken DNA, what will happen? DNA repair machinery will come into the action. It will do its magic over here and it will again realign this DNA. And that is how DNA broken, double strand broken DNA is repaired. So similarly, this DNA repair machinery may misidentify this end and this end as they are open end, they consider it as a broken piece of DNA and try to attach it with another DNA's open end. And so what will happen? This normal DNA will become a damaged DNA. Why? Because it is mistakenly attached. But because as we have here telomeres, and these telomeres bind with one very important protein that is called as a shelterin protein complex. Once telomere binds with the shelterin protein, let's say this is the shelterin protein and telomeres bind with it, then what will happen? Our DNA repair machinery will now properly identify this terminal part of DNA that it is not a damaged DNA, rather it is a normal terminal part of the DNA. So that is how this protective cap function of telomere is in the action. The second important function of telomere is that it solves one very important or very unique problem which is called as a end-replication problem. End-replication problem. So let's first understand that what is end-replication problem, and then only we can understand that how telomere solves this end-replication problem. So to understand about this end-replication problem, again consider that this is the human linear DNA. So there are two strands. Again, let's say this is the 5' end, this is the 3' end, and this is the 3' end, and this is the 5' end. Now, when this DNA undergoes replication, we know that DNA, while synthesizing new DNA, template strand is read from 3' direction to 5' direction. And the first piece which is going to be incorporated is the RNA primer. So let's say this is the RNA primer. Right? Once RNA primer is in place, then only DNA synthesis starts. Right? So this is the new DNA synthesized. Similarly, on the opposite strand, at the 3' end, first RNA primer will be inserted and then new DNA will synthesized. Now you know the concept of leading and lagging strand, right? So in a lagging strand, there will be intermittent RNA primers are also there. But what happens, these all intermittent RNA primers, they are later on removed and replaced with the proper DNA. Except for this terminal RNA primer, this one and this one. These terminal RNA primers, they are later on removed. So here both RNA primers are removed, but unlike this interior RNA primers, they are not replaced with the DNA. So what will happen? This part is missing and this part is also missing. So what happened? This blue part, which represents a parent DNA, and this black line, it represents a daughter DNA. And if you compare the length, see, this parent's DNA's length is this much, whereas daughter DNA's length is only this much. So we can say that with each replication, DNA gets shorter. So this is a problem of replication and DNA is shortening from the end. So this particular problem is called as a end replication problem. Now let's understand that how telomeres solve this end replication problem. So telomeres doesn't stop this DNA shortening, but what telomeres does? Telomeres make sure that whatever DNA is lost over in this, whatever DNA is lost, it is not of the coding region. Rather, it is a DNA of telomere itself. So in other words, you can say that telomeres are sacrificing themselves to save this coding region, right? So that is how this end replication problem is solved by telomere. Now with each replication, approximately 50 to 200 base pairs are lost. So you can easily imagine that if this DNA undergoes replication, then daughter DNA will be shorter. Once again this shorter DNA undergoes replication, it will become further shorter. So with each replication cycle, DNA undergoes shortening and shortening and shortening. But till what point? Till the point where telomeres become critically short. Critically short. When we will consider telomeres as being a critically short? When telomeres are not able to bind with the shelterin protein complex. At that time we will consider particular telomere as a critically short telomere, right? So for this, initially we have longer telomere, but with all our subsequent replication cycle, telomere undergoes shortening. Until what point? Until it becomes a critically short. So that number of replication which required for the telomere to become a critically short, it is called as a Hayflick limit. So after Hayflick limit, telomeres become critically short and they will not able to bind with the shelterin complex. Now we understand that if telomere, they are not able to bind with the shelterin complex, what will happen? See. What will happen? Now this shelterin complex are not there. So what will happen? Our DNA repair machinery, DNA repair proteins or machinery, they will misidentify this part of DNA as a broken piece of DNA, right? So what will happen? Once this telomere become critically short, our cell recognizes such DNA as a damaged DNA. And this leads to very complex sequence of event, multi-step process, which ultimately leads to a special state which is called as a cell cycle arrest. It is also known as a cell senescence. And such cells are called as a senescent cells. This senescent cells has a certain characteristic. The first and very important characteristic is that they are not able to divide. And as they are not able to divide, they cannot regenerate. Second, it leads to a tissue dysfunction. Tissue dysfunction. The third important aspect is that such senescent cells secretes a pro-inflammatory factors. So all these three factors in combination, it leads to a cell dysfunction. It leads to all the typical characteristics of the aging. We know that in the aged people, there is a loose skin, their elasticity is lost, their vascular walls elasticity is lost. This all signs of aging are because of this characteristic of the senescent cells. And ultimately, these senescent cells within a short time undergoes a programmed cell death, which is called as a apoptosis. So cell will die. So now you understand that what is the role of telomeres in the aging process. The longer telomere means the cell is young, and shorter telomere means cell became old. So length of telomere is like a molecular or mitotic clock. Okay, so it determines that whether cell is young or old. Now let us understand about the telomerase. So what is telomerase? Telomerase. So as you can see, it is ending with the word ASE. That means it is an enzyme. Telomerase is nothing but the enzyme. And what is the function of this enzyme? This enzyme, it regenerates telomeres. So it can increases the length of telomere. Now this enzyme, it is made up of two things. One is the protein and its prosthetic part is the RNA molecule. This RNA molecule as a part of enzyme, it acts as a template to synthesize DNA, right? We know that telomere is a part of DNA and it is being synthesized by this enzyme. And obviously this enzyme needs a template. So that template is within this enzyme itself in the form of RNA. So by using the information on its inbuilt RNA, it will synthesize DNA, which is the telomeres. So we can call that this is a reverse transcriptase. It is a type of reverse transcriptase. And remember, the telomerase is absent in most of the somatic cells of the human being. It is present at only few selected places. What are these places? The first one is germ cells and second one is the stem cells. Remember, as telomerase is increasing the length of telomeres, that means telomerase will prevent any cell to undergo old age. It will keep particular cell forever young, right? So germ cells and stem cells, they contain telomerase. So these cells will remain forever young. Now why it is necessary for these cells to remain forever young? See, germ cells means sperm or ovum. And these cells, we know that these cells needs to go infinite replication and infinite division, so that the genetic data is preserved from one generation to another generation, right? So that's why this telomerase is active in the germ cell. Similarly, in the stem cells are the cells which can regenerate the damaged tissue, damaged cells, right? The new cells synthesized from the stem cells will take place of the old cells. So for the stem cells also, they need a infinite replication power, infinite cell division power, and that is provided by the telomerase. So now I think that I can read your mind. You must be thinking that for forever to be young, we need telomerase, right? Or in other words, you may think that why Mother Nature had not given us telomerase in all the cells rather than the selected cells. Why not in all the cells? So we can be a forever young. Or you might also think that modern day biochemistry or modern day genetic is so advanced, so powerful that we can activate this telomerase in all the cells and we can become forever young. But remember, telomerase is obviously, it is a hero, but don't forget it is a villain Also, it is very big villain. So consider telomerase as a double-edged sword. It is not only hero, but it is also a villain. How? See, just to give you example, suppose this is the somatic cell and this is a normal somatic cell where telomerase is not active. Telomerase is absent. Now somehow this somatic cell's DNA is damaged. So what happens? This cell undergoes uncontrolled cell division. And remember, such DNA damage are so frequent in our body, it is occurring all the time. Some cells are undergoing uncontrolled cell division all the time. So what will happen? This cell will divide multiple times, right? So there will be a creation of microtumor. But this will not advance more than microtumor. Why? Because this cell undergoes so many cell division in such a short time, so that telomere undergoes a critical shortening, and so these cells will start to die. And so this tumor is never manifested in our normal day-to-day life, although every day so many cells are undergoing uncontrolled cell division. So their fate is death. Why? Because telomeres are gone. But what will happen if telomerase is being active somehow? Then what will happen? The death of cell will not occur and this cell will become immortal. So tumor will become more and more larger and ultimately it leads to cancer. Right? So remember, by being absent telomerase in the somatic cell, we are saved from the cancer. So it is actually a defense mechanism against the cancer. And in real life, 80 to 90% of cancers, if you take their cells and examine, their telomerase activity will be present. So remember this very important point, that telomerase, don't consider it as just a hero. It is also a villain and very big villain, right? So now I hope that you understand both the term telomeres as well as telomerase and what they do. And you also understand that how they are involved in the aging process. Just to recap, if longer telomere, that means cells are young. If shorter telomeres, that means cells are old. And telomerase, if higher telomerase activity, particular cell will remain forever young. If telomerase activity is no, then that cell will slowly become older and then death will occur. So that's all for this video. I hope that all the concepts are clear. The model answer is given in the PDF and the link to download this PDF is given in the description of this video. If you have any query or confusion, please write it down in the comment section below. Thank you.